kind of topics which is you don't need to memorize a lot of things because everything's right, you can think in a logical way. Yeah, so this is inheritance. So what's meant by inheritance? Inheritance is they refer to the transmissions of the genetic information from generations to another generations. So when we talk about inheritance, it's talk about the DNA. Okay, where you can find the DNA. DNA is the trap-like structures. So DNA is found inside the nucleus, right? So you can see this is a nucleus. So in the nucleus, there will be something like this. Okay, let me draw a nucleus for you. It's very bad. My pen is not functional. Okay, it's something like this. I'm actually drawing nucleus to you. This is a nucleus, right? Okay, I draw in this way is because at nucleus they have a double membrane. Nucleus is double membrane, just like in the mitochondria and chloroplast, they have the double membrane. And then in the middle parts, right, they have the nucleus. Nucleus, okay, which is this one. A large prominence black spot, okay, at here. Okay, here is a nucleus. So what is the structure which surround the nucleus? Is the DNA, okay? So you can see the DNA, it will be like this. It look messy. To be honest, it really look messy. So this is the DNA. DNA is the thread-like structure, which is enclosed within the nucleus. So what is the function of DNA? What the DNA do is they will carry the genetic information in form of genes. So at this way, I'm not sure, do you able to differentiate what is meant by genes and what is meant by DNA? When we talk about DNA, DNA is something like polymers, you know? DNA is a kind of polymers, just like polysaccharide, just like polypeptide. DNA is actually a polymer because when we talk about DNA, right? When we talk about DNA is like this, okay? You can imagine this is a DNA, okay? DNA is like this, right? Like this. It's double-stranded DNA, okay? No ending. If you view the DNA under microscope, right? It's not microscope. Under the electron microscope, you can see DNA is like this. And then DNA usually is no ending because it's very long. It's super long. And then there's two strands, right, intertwined to each other. So they will form what? They will become double helix. Or double strand. So this is the DNA, okay? Then what is meant by gene? Okay, it's because that DNA is very long, right? And then this DNA inside, they contain what? DNA, they will store the genetic informations. They will store the genetic informations inside. Okay, so what's meant by genetic information? It refers to this genetic information, they can do what? They can help to produce proteins later. Used for protein synthesis. So this is functions of the DNA. Okay, I put it here. Yeah, used to synthesis for the proteins, right? And then the genes is they refer to a portion of DNA because DNA is super long, right? And no ending. So the genes is they refer to here. So when you look at these portions, here to here will be one gene, okay? For example, gene A, no, gene A. So later, this gene A, right, they will undergo the transcriptions. So later, they will do the translations, and then they will become what? At the end, they will become the proteins A, for example, protein A. And then you have another genes, which is because what? Gene B here. another portion of the gene. So you can see, right, it's actually located on the same strand of DNA, but at the different locations of the DNA. So this is gene B, for example. Gene B, okay? Gene B. So what gene B do? Okay, gene B later, they will also undergo the transcriptions and translations, and then they will become proteins B. Protein B. Okay, so if you want to give, for example, gene A is like a genes, okay? A genes, they will decide your hair color, you know. Your hair color. So the protein will come out, right? It will become what color? Something like brown color. Okay, brown color of the hair. Then how about gene B? Gene B, something like it's a genes. They will decide your eye color, okay, your pupil color. So at the end, protein B, right? What they will show? They will show the color of pupil, which is, for example, the black color. So, at that proteins is just the expressions of the gene. It's because that genes contain the genetic informations, they will be used up to produce proteins later. So you know the difference of the DNA and gene. DNA is referred to the whole strands, very long, very complex, no ending. Genes is only referred to the portion, the short portions, or we call it a length of DNA. Just a short length of DNA, which is they can use to encode to make proteins later. Yeah, so you can see that a gene is referred to the short length of DNA found on the chromosomes. Okay, they code for a specific proteins. So it's because the genes, they contain the specific information. So those specific information, they will undergo the transcription and translation later in order to translate that become the proteins, structural proteins. So you already know that the difference of the genes and the DNA. Then how about chromosomes? The chromosome is referred to the condensed form of DNA. It's because that you can have a look, right? This is the DNA, right? Okay, DNA, you know that DNA is long strands. So what happens if the DNA, they coil? They just coil together, okay? Then they become a butterfly-like shape. So it will become chromosome. Chromosome in Hawaii it calls what? It calls a 染色体. Right? Something like this. See? And then the question is, why the DNA need to condense itself, become a butterfly-like chromosome? Why? It's because later the chromosome, they will need to do the divisions later. They need to undergo the divisions in the mitosis, right? They need to undergo the anaphase. Yeah. So that's why it's better for them to coil in this butterfly shape. So it's easier for them to undergo separations of the sister chromatic later.
I have a question.
Sorry?
I have a question about the chromosomes.
Yeah. Yes,
Why would having one extra or one less chromosome be bad?
Sorry, your voice is a bit blur. Or you want to type?
Like why
I
would
think you can type. Why?
like Down syndrome, right? Down syndrome
Down syndrome, yes.
caused by chromosome. So how does that happen?
You mean how the chromosomes result in the Down syndrome?
Yeah.
Okay, Down syndrome is very interesting case. It's because of what? I let you see a picture, right? The Down syndrome is like this. Down syndrome, you have extra one chromosome. Down syndrome is due to the failure of separation of the sister chromatic, which it result in the extra copy of chromosome. So it becomes 47 chromosome instead of 46. So let me show you a picture. Down syndromes. The chromosome, okay, the chromosome. So the reason is due to the failure, okay, failure of the separations of the chromosome. See, Down syndrome is being caused by the failure of the chromosome being separated properly during cell divisions. So this is known as what? Non-disjunction. Okay, actually this is not included in your syllabus, but I just let you know. Okay, I'll just keep you the picture. So like this. So you have, maybe I can put here. I think I can put here, right? So we call it what? We call it trisomy 21. Why we call it trisomy 21 is because that this kind of non-disjunction is happen at the chromosome number 21. You have how many chromosomes in your body cell? And that's where you have how many chromosome number? How many chromosomes? Or I mean, how many pairs of chromosomes inside in the every or body cell? How many pairs?
23.
Sorry? 20?
23.
Yeah, 23. You have about 23 in every of your body cell. I refer to your somatic cell. Somatic cell, okay, you have 23 pairs, which is in total would be 46 chromosomes, right? Yeah, somatic cell. Somatic is, I refer to the body cell, okay? I refer to the body cell. So what happens for the Down syndrome is like the egg cell and the sperm cell, right? Okay. The egg cell and sperm cell, okay, what happened is, for example, and then this is usually that occurs from the mother side, you know, from the mothers. So especially the Down syndrome happens is because that the mother is already, it's already not in their suitable fertile age. I mean, we call it 高龄产妇, you know, 高龄产妇is referred to the women, right? They give birth in the such a later age. Maybe they reach about 40 years old ago. I mean, yeah, 40 years old above, they will still give birth. So they will have this kind of risk of the Down syndrome. So you can see, right, the egg cell and the sperm cell. So initially, okay, this is, they refer to their parent cell, parents of the egg cell and the sperm cell, parent cell. Because their parent cell initially is diploid, okay, it's not haploid, it's diploid. How can I? Just you need to know that this is the parent cell. Okay, I just put P at here. P. Okay, this is the parent cell. Same to the sperm also. So after that, the egg cell, right, they need to undergo what? Meiosis, right? They need to undergo meiosis, which is this process. Okay, meiosis. Meiosis. Yeah, same to the sperm cell, they also need to undergo meiosis. So after the egg cell undergo meiosis, they will become 23 chromosomes. But the problem is there is about one copy of chromosome, right, failure to undergo separations. So instead of they become 23, they'll become 24. So you know, because in the normal, in the normal chromosome, right, it will become 23 for each. But if you have the failure of separations of the chromosome, right, it will result in 22. So it means that this 22, right, it's because that there's a one copy of chromosome being contribute to the normal chromosome. So if you sum up, become 24 chromosomes. So the concept is something like this. I think I'd show you a picture better. So you can see this is because 47 chromosomes in every of our body cell. You know, it's not only one body cell. Every of our body cell also have this 21 chromosome. So that's why the Down syndrome, right, the children will appear in the, a normal appearance, okay? And then they will have a lot of like defects. So let me show you this one. So this is due to the non-disjunction happen, okay? Non-disjunctions, right? Do I have a better picture to show you? I think this is a great picture. Okay, this one. It's a nice picture to show you. Okay, non-disjunctions. Non-disjunction during meiosis, okay? Disjunctions during meiosis. Okay, I think I better use this picture. Okay, so you can see, right, what happened in the meiosis. This is in the normal separations. In the normal separations, you have the chromosome, right? You have about a chromosome, okay, a pair of chromosome. So after you undergo the normal separation, right, you become what? And then, you know, meiosis, actually there's two phases. There's two phases of meiosis. It's not about in mitosis. Mitosis, you only have one time only, but in meiosis, you have two phases. First time, you are actually separate the homologous chromosome, you know, homologous chromosome, which is another pair of chromosomes. This is the first, what happened in the first meiosis, okay? You are actually separates what? Separates the homologous chromosome. Okay, you will learn about this homologous. What is meant by homologous chromosome? Homologous chromosome is that actually refer to the chromosome which is have a same length, same position of the centromere. So this is known as homologous, which is when you draw about this chromosome, right? It will be something like this, okay. And then this will be another pair, okay. So one is from the father, one is from mother. So this homologous chromosome. So you can see, right, same length, same position of the centromere. You can see, right, also at here. So this is called as homologous. So actually in the meiosis 1, right, in the first phase, you actually separate the homologous chromosome. But what happened in the second meiosis 2, right, you actually separate the sister chromatic. You actually separate here. You can see, right, here, like this, like this. You separate the, we call it sister chromatic, which is on the same chromosome. Sister chromatic. But what happens in the meiosis, during the meiosis divisions, is because that the non-disjunction happens. So you can see, right, the non-disjunction happens instead of like in the normal stage, right? In the normal stage, you can see the sister chromatic already be separated, so become 23 for each. So due to the non-disjunction happen, right, it will cause the two sister chromatic, right, directly go to become one at here. Okay, which is, so they'll become 24. And then here will become 22. They become 22 because there's no more chromosome inside, so they'll become 22. So this 24, right, if you fuse with another gametes, okay, if this one you fuse with another gametes, which is 23, right, you become what? 47. So this 47 will be result in the Down syndrome, because this 47 is a normal number of chromosomes. We shouldn't have 47. So this is called non-disjunctions. Non-disjunction is happens due to the chromosome itself, they just lost its elasticity. So they cannot separate. It's not, it's referred to this one, okay? It's referred to spindle fiber, you know? The spindle fiber, they just lost its elasticity. You can just imagine, right? There's a spindle fiber being produced during the anaphase. Okay, yeah, during the anaphase. So there will be something like this. This is a fiber, right? Yeah. So one fiber will pull from here, another one will pull from here. So why the non-disjunction happen is due to the spindle fibers, right? They just lost their elasticity. They lost their function. So they are unable to pull, they are unable to separate the chromosome apart. So they will result in the extra copy of chromosome being carried by one of the gametes. So when this extra copy of chromosome carried by one of gametes fused with another normal gamete with 23, right, they'll become 47. So adults, are you clear about this?
Yeah.
Yeah, so non-disjunctions. Usually it happens due to the female. Female is very old already, but they still want to have child, so that's why they will have become high risk for this. Yeah. And then what I need to tell you. So you can see that chromosome, okay, this is chromosome. So in humans, okay, in the normal humans, right, our chromosomes number will be 46, okay? So this is our human, we call it human karyotype, okay, karyotype. How many? It's about 46, okay, 46. So we have about 23 pair, okay? Like this, 23 pair. So maybe I can put here. Oh, too big already. So this 23 pair, right? 22 of them is belong to somatic cell, which is the body cell, okay? Another one pair will belong to the sex cell. So you see, you have about 22 pairs, okay? So it's all belong to the somatic cell. 22 pairs of chromosome, right, is somatic body cell. Okay, Adam, so tell me, somatic body cell, they undergo mitosis or meiosis? They undergo which type of divisions? Meiosis or mitosis? Tell me. Hi. It's meiosis or mitosis? So yeah, Ethan? It's okay, you can just guess one of the answer. Meiosis or mitosis?
Meiosis.
Oh, okay. Just I asked about one. Oh, I asked about this one, right? It's mitosis. It's mitosis. In the somatic body cell. Okay. How you able to differentiate? This is you refer to this one. Okay. You refer to this. Oh, here. I think it's here. Yeah. Okay. You can have a look on this one. Yeah. The purpose for the meiosis and the mitosis. So you can see my screen, right? I'm showing the Google. I'm showing the Word document.
Yeah.
Okay. So meiosis, their purpose is they will do for the sexual reproductions. 有性繁殖, okay, sexual reproductions, which is the purpose of meiosis is you want to produce the gametes, the sex gametes. Okay, for example, is the sperm, the ovum. So it's something like this, okay? This is the meiosis. So you can see, right, initially the meiosis, which is their parent cell. The parent cell also have the diploid. Diploid means that they have two cell chromosomes, right? But after they undergo meiosis, they become what? They will produce 4 haploid cell, which is the daughter cell. Yeah. So this is the way on how your body produce the ovum and also the sperm cell. Also by this way, meiosis. Okay. Also by this way, meiosis. Because meiosis, right? Meiosis is different from the mitosis. It's because that meiosis, right? Okay, which is they involve in separation. You know, they involve separation of the sister chromatids and also hormonal chromosome, which is just like I show you. Meiosis, right? Their process involved in the separations. Okay, this one involved in the separations of the homologous chromosomes and the sister chromatid, which is just like I show you in the picture, right? So that's why at the end you produce the daughter cell, which only have half of the chromosome. So only you have about 23 instead of 46. Just half a chromosome. Any of the cell, if after they undergo meiosis, right, they only have half. For example, if initially their parent cell is about 96, so if you divide to 2, become what? Oh, sorry, I also don't know. 96, I divide to 2, it become 96 is not a good example, okay? Because it will have it belong 48. It belong 48. So remember, meiosis will produce the 4 haploid cell, and each of the haploid cell have half of their original chromosome number. So do you know why they need to have half of the number? Why they need to have half a number? It's because that meiosis later to do what? They will fuse back together, right? Because they have these 6 gametes, okay? They will produce these 6 gametes, which is the sperm, the ovum. So later the sperm need to fuse with the ovum so they can restore back the diploid number. So that's why the ovum is 23 chromosomes. Then the sperm also, they have 23 chromosomes. So that's why you mix them together, you sum up together, become 46, which is the diploid zygote. Yeah. So later, diploid zygote, what they do is they will do the division. Okay, they will do the divisions, which is we talk about mitosis. So remember, meiosis is only occur in the 6 gamete cell only. Okay. So example of the meiosis, right, is what is the sperm, ovum, and also the pollen grains. Okay, pollen grains. So you need the fusion of the 2 gametes, right, to form the diploid zygote. So later the zygotes, after they restore their diploid number of chromosome, they will undergo mitosis to increase number of cells. So how about the purpose of mitosis? Okay, mitosis is just very simple, okay? Because mitosis, right, is just about from 1 become 2, right? From 1 become 2. So, and then their daughter cell, their chromosome number is totally same with their parent cell. Which is, if their parent cell have 96, their daughter cell also have 96. That's all. Okay. So because the purpose of mitosis is not produce 6 gametes, okay. In the meiosis is because they want to produce 6 gametes, which is haploid cell, in order to restore the diploid number of chromosomes later after they become zygote. But in the mitosis, right, what we want to do is you actually do what? You actually increase the number of cells, especially increase the number of the body cell, your somatic body cell, for growth, for tissue repair, for asexual reproduction, like in the binary fictions, right? Okay, so I hope you are clear, okay, about the process. I mean, not process, the purpose for the meiosis and the mitosis. So just now I talked about this somatic cell, right? So actually, this somatic cell, all of these 22 pair of cells, is they will undergo cell divisions later, which is the mitosis, okay? Mitosis later. Then how about this XY? Okay, this XY, right, is the 6 gametes, okay? It's the 6 gametes. So 6 gametes, right? They will do what? They will gametes. Okay, later, they will, it's being produced, okay? It's being produced by the meiosis, okay? Yeah, that's all. So that's why when you want to describe about the request human karyotype, right? Usually they will just write in this way, okay? For the male, will be what? The male, right? It will be the 22, okay? 22 X plus XY, okay? XY. And the female will be female 22 pairs plus XX. Yeah, so this is how you can write. So total will be about 46. Yeah, it's about 46. Okay. So yeah, so there's a part for here. So the function of genes, right? Okay, genes, since they can code for the proteins. So actually what are found inside the genes is genes, they actually contain the characteristic. They actually can help to decide the cell characteristics, their functions, their structures. They are all being decided by the genetic information, which is they are found inside the genes. Okay, that's all for this one. So remember the difference in the DNA, the long strands, large and complex strands, right? And then the genes, okay, the gene just a portions, just a short length of the DNAs, and then chromosome, which is the butterfly-like shapes, due to the long strand of DNA, is being condensed, fully condensed together with the proteins. I'm not sure I got put in this notes or not, which is for this one. Okay, I didn't put, I think I should put, sorry. I think I should put a picture, okay, to help you to clarify better, which is this one. The difference, okay, the difference for the genes, right? Genes and the DNA and the chromosome. So you can refer to this one. This is a very nice picture, right? Okay, nice picture. I should put this one. See, so the chromosome is due to the DNA. They already condensed with the proteins, right? Actually, the DNA is a long strand. If the DNA want to condense become chromosome, right? DNA need to associate it with the histone proteins. I'm not sure you've heard about this term before or not. Histone proteins is referred to like this. Okay, which is this one. DNA associated with the histone proteins to produce a chromosome. Okay, to produce a chromosome. So yeah, it's something like this, right? So this is DNA. Okay, DNA with the base pair. So the double helix, right? They need to associate with the histone protein. So you can see, right? This is histone protein inside. Yeah, so this histone protein. So after that, they will condense, and then they'll become the chromosome, which is the butterfly-like shape, right? With the centromere at the central part. And then they are found inside the nucleus. The end. So the nucleotide base sequence inside the DNA, later they will do what? Nucleotide base sequence inside DNA, right? Later they will undergo the, we call as what? We call as a transcriptions, okay? See, transcriptions. Transcriptions is a kind of process which is they will convert this. Actually, it's a process to make the messenger RNA from the nucleotide base sequence on the DNA. So because messenger RNA, right? This mRNA, we call it messenger RNA, it's just a copycat. It's just a copy of the portion, okay, of just a portions, copy of a portion of the genetic base sequence on the DNA. Because when the DNA, right, they want to start to produce the proteins, they will start to uncoil themselves. But it's not whole DNA strand will uncoil, just a certain portion only. So that's why this portion, right, is actually known as the gene, okay, the gene, which is a short strand, okay. I mean a short length of the DNA, right? They will unwind themselves. So that's why they can have the enzyme. So the enzyme, they can start to produce a messenger RNA. So how they can produce a messenger RNA is they will start to form the base pairing, okay? We call it as a complementary base pairing. They will have a complementary base pairing between the DNA and the messenger RNA. So you can see, right, this term, oh, what happened? You can see, right, the picture, right? The pairing between of the A to U, C to G, A to U, C, G, yeah, this kind of pairing. Okay, so if the question asks you, so you can have a look. If the question asks you, okay, why is it important that pollen nuclei are haploid? Why pollen nuclei didn't really haploid in number, just like in the sperm or in the ovum? It's because that they need to restore the diploid number after they undergo fertilizations. And then why the sperm and egg cell are haploid but not diploid? It's because that they want to restore, which is they restore the diploid number of chromosome after they undergo the fertilizations. And also, they cannot become diploid number, right? So it's because that they want to prevent, okay, to prevent diploid number of chromosome does not double at fertilizations. So that's why they need to undergo meiosis to separate their chromosomes, separate their sister chromatic. That's the story, okay? And then we continue. So this is the description, okay, for the genetic inheritance traits, dominant recessive trait. Okay, what does this mean? Okay, genetic, you already know. Genetic is just a study of the inheritance. Okay, just study of inheritance. It's study about how variations, right, arise and how the character individuals are passed on. And then inheritance just passing down the instructions from generation to the next generations. Then if they talk about characteristic, the characteristic is they just refer to the requires like something like your eye color, your skin color, or even your height, your weight, which is your characteristic. They become you, okay? They become you. The trait, right? The trait is actually they refer to, we call as what? Phenotype, okay? I'm not sure. Yeah, phenotype. And also, have you heard of this equation before? This is very important equation when you study inheritance. Okay, this is not found in your textbook, but I just want to show you. It's something like this. You will have the phenotype, right? Phenotype equal to the genotype, genotype, and plus the environmental factor, environmental factors. So what is mean by this? Okay, you can just imagine, right? Phenotype is the physical expressions of that gene. Genotype, it just refer to the gene, okay? It just refer to the gene. Okay, the genes, right, we call as genotype. Either it's something like, you know, if you are tall, okay, for example, if I want to describe whether the tall gene or short gene, okay, if I tell that the tall gene, okay, the tall gene is something like this, big A, small a, or 2 big A. And then the short genes, it become 2 small a. So the genes, right, are referred to this one. Either is tall gene or short gene. Okay, so this is genotype. Yeah, this is genotype. And then how about environment factors? So environment factor is any kind of factors that can influence the height of the person in their development stage. So the factor will be the nutrition or the exercise. So for example, like even though the persons, they carry a tall gene, but it's because that they don't have enough nutrition supplied to them. And also they didn't do exercise, okay? They didn't do exercise. So that's why even though they have tall gene, right, they are unable to express their tall gene. So they will still remain short from the phenotype. Okay, so this means that the environment factor, they can express your gene expressions. Oh, sorry, I mean the environment factors, they can affect, okay, they can affect your gene expressions. So the gene expressions, right, is shown in the phenotype. Yeah, so here, so it's something like this, okay? And then now we talk about traits. So the traits, it just refer to like the phenotype. Okay, phenotype is a characteristic that you can observe from the eyes. Okay, whether this person is very tall, very short, fat, or slim, right? So for example, you have the dominant trait or recessive trait. So this is just refer to what the genotype. Okay, so the dominant trait is right. This genotype, right? They have the they have a, I mean the big T. Okay, big T, or 1T, small T. So this big T, right, is referred to, they are like yeah, they're very tall. Okay, just very tall. Show that it's very tall. All gene. Okay. Then how about this one? Recessive trait, right, is referred to when they are expressed with the recessive alleles, it's paired with another recessive allele. So 2 small t. So this 2 small t, right, it will be what? It will belong to the short gene. Like this. So we can have a look on the how the chromosome look like. So for example, this is the homologous chromosome. So what is meant by homologous chromosome? I think you first time heard it, right? Homologous. Homologous refer to, like, this chromosome, they have the same locations of the centromere, which are here. This and this. Just parallel to each other, right? Same locations. And also they have a same location of the gene. So you can see, right, this gene, okay, they just pair up with another gene. Yeah. We call it some I mean gene located at the same loci. You know, the positions of the chromosomes with the gene located on it is called as loci. Loci, gene loci, loci. Gene, loci, or locus. So you can see, right, there will be different genes, okay, different genes. So you can see some of GRT. So they code for different proteins. So they will be encoded for different proteins. So at the same chromosome, right, same chromosome, they will have different gene on it. So you can see this is same chromosome. They will have different gene like the eye color, blood type, hair color, or the growth. So genes is referred to the basic unit of inheritance, which they will occupy a specific positions, we call it loci. Okay, see, loci on the chromosome. So a section of DNA, right, they can code for the particular proteins. And then the genes, right, they can help to determine the characteristic of an organism. Yeah, so this is just function of genes. Because genes, okay, and that's what's meant by genes. Genes, right, refer to these genes later, they can express, become a real protein. So this is not genes. Because not all, not all, on the DNA strand, right? On the DNA strand, okay, not all of the DNA regions they can express become the proteins, you know. Only some part they can undergo the translation and translation, and then they'll become the proteins. So the regions which is the DNA, they can undergo the translation become proteins, they know as a gene. They know the genes. And then how about this one? Alleles. Okay. Alleles is referred to the alternate copy of the same genus, same genes occupy the same reactive locus on the certain pair of the homologous chromosome. So, okay, you can highlight on here. A gene must have at least two forms of alleles. What does it mean? Okay, alleles is they refer to this one. Right, just how I show you about this. So you can see, right, the big T, two big T, one T, one small t, and two small t. So this is referred to the alleles. If you want to form a one gene, right, you need to have at least two alleles. One gene, a gene must be at least have two alleles, which is one big T, one small t, or two small t, or two big T, like this. So this allele, right, later they will determine the traits of the characters, which is later they will show you in the gene expressions. And then you have dominant alleles, you have the recessive alleles. Dominant alleles is they refer to the traits, right? They have a big capital letter, big T. Okay, recessive allele they have a small t like that. Or you can use other letter also can. Just means that dominant alleles is always a big capital, and recessive allele is a small capital. Dominance is not means that it's good or it's bad, you know. Dominance is referred to this trait will always show later, will always like presence and suppress the effect of others alleles, which is the recessive allele. So because dominant, right, they will tend to express themselves later. Then the recessive alleles, right, because is due to that the alleles is recessive, so it will be suppressed by the dominance. So it may or it may not express themselves later. It will only express themselves if they pair with another recessive alleles. So in Huai it calls what? 显性基因, you know? Dominant alleles we call it 显性. Always appear, okay? 显性. This one. Recessive alleles like invisible, okay? 隐性基因. This one. So this is just a term, okay? For your exam purpose, you need to know the genes, how you're able to describe the genes, okay? This one, the gene. Other like alleles or the traits, like characteristic, the exam will not come out. They only ask about gene only. So far, what I see, they only ask about genes. That's all. And then now we'll talk about the diploid and haploid. So what's meant by diploid? What's meant by haploid, right? It's because that the diploids, they have two sets of chromosomes, right? They have two sets. So in total, they will have about 46. So we call it diploid. Okay, see, two sets of chromosomes we know as diploid. I think I better let you see the picture of diploid and haploid. Wait me for a moment. Diploid and the haploid cell. Okay, see diploid and haploid cell. So we can see their structure is something like this. So we have a look. See, here is a diploid. Yeah. I put it here. Can you see the haploid cell and the diploid cell? So this haploid cell, there's only 3 types of cell. Okay, it's your ovum, sperm, and pollen grain. They only has 1 set of chromosome. Ovum, sperm, and the pollen grains only have 1 set of chromosome. Then how about in the diploid cell? So you can see, right? They have about two set of chromosomes. So you can see 1, 2, 1, 2, 1, 2. 2 set of chromosomes for each so-called diploid. So every of your somatic cell body, right? Somatic cell body, they are known as diploid cell. So usually diploid cell is due to the result of the mitosis, okay? The haploid cell is due to the result of the meiosis, the product of the meiosis. Okay, nuclei with 1 set of unpaired chromosomes. So you can see, right, this chromosome is unpaired. You can see unpaired. Just only 1 cell with unpaired. So called as haploid. Unpaired. So this unpaired chromosome, right, is due to the what? Due to the result of meiosis. Okay, result of meiosis. Result of meiosis. We're going to separate the homologous chromosome and also sister chromatin. So at the end, you get this 1 cell from initial 2 set. So haploid just half the number of chromosomes. And then now we talk about inheritance of 6. So I think we can do some questions. It's better for us to do some questions so you can understand more. Okay, sex or gender is determined by their entire chromosome pairs. So this is mean by the sex gametes, right? Which is they refer to X gametes or the Y gametes. So they will have this, okay, 6 chromosome. So female XX, male, they have the XY. So as only father they can pass on the Y chromosome, right? He is responsible for determining the gender of the child. And then for this one, male and female karyotypes, right? So it just difference in their sex chromosome. Other than that, it's all the same. Okay. They also have 22 pairs. 22 pairs. Just like male, they have 1 X, 1 Y. And then the female, they have 2 X. And then adults will have a look. You can have a look on this chromosome. X chromosome and Y chromosome. You tell me which chromosome has a larger size? Which one has smaller size? Which chromosome have larger size? Which one has small size? Is X chromosome or Y chromosome? Which one have larger size?
X chromosome.
Yes, it's X chromosome. It's larger size. Then how about Y chromosome is smaller in size. So that's why there's a lot of required sex-linked disease. Sorry, it's a bit noisy here. Oh my God. Oh. Okay, there's a lot of sex-linked disease, you know. The sex-linked disease is happen at the X chromosome. You can write down here, sex-linked disease. They happen at the X chromosome. Okay, they can locate on the X chromosome. But not at Y chromosome. So that's why a lot of sex-linked gene is located on the X chromosome. It's because X chromosome is too large already. It's very large. They can occupy a lot of genes. Okay, so this is one thing. Okay, another thing is, yeah, so it's something like this, okay? The sperm cell. So half of all the sperms will carry an X chromosome, and half of the sperm will carry the Y chromosome. So if the child, okay, inherits as Y chromosome, it will become male. So at this point, both of you are XY, okay? Then I'm XX. Yeah, so all X cells, right, they will only carry X chromosome. So that's why the sex of the child, right, is actually determined by the father, because only father they can pass on the Y chromosome, which is a smaller chromosome. Okay, the smaller chromosome. Okay, so this is sometimes the exam they will ask you to write this kind of genetic diagram called as Punnett square. Okay, Punnett square is just like you put something like this. Okay, this is the gametes from mother and father. There will always be, there will always be two copy, right? So mother is SS, father is SY. So this is the offspring. So this is a possibility combination of the offsprings. So at the end, you produce about 50% of them is boy, 50% is from the girls. Yeah. And then now we talk about transcription and translations, right? The transcription and translations. I think you heard of this before, right? The first step, if the DNA, right, the long strand of DNA, they want to produce, they want to undergo the translation become a protein, right? First step is they will need to unwind, or you can say uncoil, which is that regions of DNA. Initially is like this, okay, it's intertwined, right? Initially is intertwined. So after you unwind, that means the regions open up. Okay, you can see, right? The regions open up. Like this. Like this. So actually, this region, which is they open up, right? They actually is a gene, okay? Because you can open up this gene, right? And then you can synthesize the messenger RNA. So messenger RNA, they can bind to ribosome and then they can start to produce the protein. So you can see this is the DNA. Okay, DNA is like here. Here is the DNA. DNA. Oh, DNA. And then here is the messenger RNA. You can see, right? Because RNA, the difference of the RNAs is because the base, okay? In the RNA, you don't have thymines. You know, you don't have thymines. In the RNA, the thymine is being replaced with the uracil, which is U. So that's why the A only can pair with U, not T. So this is the RNA, okay? Which is the messenger RNA. RNA. So after the gene is copied into messenger RNA, so messenger RNA, what they do, and then messenger RNA is shorter strand, is much more shorter strand. So that's why messenger RNA, they can come out. They can come out from the nucleus and get into a cytoplasm because their strand is shorter. It's much more convenient for them to travel by the, to pass through the nuclear pore and get into a cytoplasm. So you can see that in the third step, messenger RNA, they just move out from the nucleus to the cytoplasm. And then they will bind to the ribosome. Let's say, see, here's the ribosome. And the ribosome start to join the MSC together. They'll become the polypeptide chain. So you can see, right? They start to form polypeptide chains. So after that, the polypeptide chain, they will fold to each other. They'll become a shape. See, they'll start to form a shape. So this shape, right, they'll become the protein structures. Either it's enzyme or antibody or hormones, any kind of protein structures. So this is the whole process. So everything just originated from the DNA because it contains the genetic information to make the proteins. Okay, so this is the whole process. This is the whole process. Okay, you can have a read. Okay, another thing is, last time I didn't show you that actually how the ribosome actually help to make the proteins. Because ribosome is just like chef, you know, the cooking chef. Because ribosome, right, is the organelle to produce a protein. It's just like the chef. The cooking chef is the one they start to make the food. Okay, how they can make the food? First, they need to have the book first. Okay, they need to refer to the cooking book. And then they need to read on the specific page on the cooking book, right? So the cooking book is referred to the messenger RNA because the messenger RNA, they just carry the information from the DNA. So after that, they need to prepare the ingredients, right? So the ingredient in here will be what? Will be the amino acids. So where you can get the amino acids? You can get the amino acid from the transfer RNA. So you can see, right, the transfer RNA at here. tRNA, we call it what? tRNA. This is the transfer RNA. So the transfer RNAs is also kind of RNA, okay? It's also a kind of RNA. So the transfer RNA, right, is directly bind to the amino acids. At those you have how many types of amino acids? You have about 20 types of different amino acids. 有20种, okay? 20 types. So what transfer RNA do is they will carry the amino acid on their head part, okay? And then they'll bring to where? they'll bring to the messenger RNA. Yeah, they'll bring to the messenger RNA. The mRNA, okay, they'll bring to the mRNA, and then they can bind to the mRNA. But you can see, a very special structure is, you can see the tRNA, right? They only bind to about 3 base. You can see the 3 base like UAC, UGG, ACA, right? The 3 base only. So this is their feature, okay? Each of one tRNA molecule, they can only bind to 3 base. So this equals what? Triplet anticodon. Triplet. Okay, and that's well. The base, okay, the nucleotide base, which is located on the tRNA, we call as the anticodon. The name known as anticodon. Then the nucleotide base, which is they located on the mRNA, right? They have a name known as codon. So this is called codon. Codons on the mRNA. Messenger RNA. So you have what? You have the anticodons from the tRNA paired up with the codons on the messenger RNA. So by this way, right, you can join the MSC together, right? Because each of the anticodons on the tRNA, right, they can join with one amino acid, one type of amino acid. Sorry, it's one type of amino acid. So you can see at this picture, right, it shows what? Valine, prolines, tyrosines. Actually, it's referred to the amino acids, the type of amino acids. So, and then how they can join the amino acid together? They join amino acid together by forming the peptide bond. Yeah, so you can see, right, here's the peptide bond. So at the end, they can become polypeptide chain. So the way, which is the anticodon from the tRNA joined to the codons on the mRNA by the complementary base pairing. So you can write about this one. Okay. If the question asks you about this, right, you can write about this. So this complement base pairing is occur at the ribosome. So we can have a look on this one. And then the reason why the cell need to produce a messenger RNA is because messenger RNA, right, is much more shorter, convenient for them to go out, because DNA is too large already. As I told you before, right, DNA is a large, it's not ended, okay, large, complex, long strands, right? So they cannot go out nucleus. They only can stay within the nucleus. They cannot go out. So that's why they need somebody to help to copy the information out to the cytoplasm. So the protein are made in the ribosome, right, with the sequence of amino acids controlled by the sequence of base contained in the DNA. So as a DNA, they cannot travel out the nucleus to the ribosome. So that's why the base code for each gene, right, is transcribed into the RNAs. So because of transcribed, okay, is this word, transcribed. This is a keyword you should write in the answer. So it calls messenger RNA. So mRNA, they can move out nucleus and then they bind to the ribosome. So ribosome, they can help to read the codes, okay, which is the codon. Okay, they have to read the codons and the mRNA. So each triplet code, so you can see, right, each triplet of the base triplets, because you have three base code for one amino acid, so it calls triplet. Okay, each triplets of the base they code with the one amino acid. So after that, the ribosome, they will translate the base into the sequence of amino acid they can make out protein. Okay, so in summary, it will be something like this. Okay, first you need to have this one, sequence of the nucleotide base on the DNA, right? So you're able to become what? Become the sequence. First become the mRNA first. Sorry, messenger RNA, right? So after that, you become what? You bind to the ribosome, right? Ribosome. So ribosome, they start to join the amino acid together. So at the end, you can form the sequence of the amino acids. Sequence of the amino acids. And then this sequence of amino acid, right, they will become what? Become the polypeptide chain. So they will start to form the structure of the proteins. So this structure of protein, right, later they'll become what? They will become the functions of proteins. do you know that the function of protein is actually being determined by its structure? So what determines the structure of proteins? So it's a sequence of the amino acids. Okay, functions of the proteins. Their shape, their structure will affect their functions. So that's why the sequence of amino acid is very important. So if you change, so for example, if you change the sequence of the base, if you just change one base position, it might cause the proteins, okay, become a difference already. It's because that if you change the base, right, you can induce a change in the amino acid sequence also. So at the end, you change the structure of proteins, and then you can change the function of proteins. For example, like the sickle cell anemia. I'm not sure you heard about this before, right? Sickle cell anemia is a kind of anemia due to the mutation from the DNA base. Okay, that's all. So just need to remember transcriptions, later translations at the ribosome. And then, okay, for this one, DNA, right? What they do is DNA can help to control the cell functions. So how are they able to control cell functions? They can control the production of proteins. So that's why I think you learned this before, right? Which is one of the function of cell is they will produce a protein. So what kind of proteins they produce? What kind of protein products they will produce by the cell? For example, is the enzyme, antibody receptor, and also like hormones, hormones, glycoproteins. They are all kind of protein product. So this protein product, right, actually they can help our body to function. So those are under the control of the DNA, because DNA is the one they can decide whether they want to produce a protein or not, or they don't want to produce a protein. So all are decided by the DNA, So DNA is a big boss, okay? DNA is a big boss. So although all body cells in the organism contain the same genes, right? Many genes in the particular cell are not expressed. So you can see, you can see this statement. Even though all body cells in the organism contain same gene, but many genes in the particular cell are not expressed because the cell only make specific protein is needed. Okay, so what does this mean? That you can just imagine, right? Like that's where you yourself, right? You are actually the products of the mitosis by the diapause-zygote, right? You are actually from the diapause-zygote, okay? This one, diapause-zygote. Initially, right, you have a sperm and ovum. They'll fuse together. They'll undergo what? Fertilization. So after they fuse together, will become what? You become the diapause-zygote. So this is you, okay? This is you, diapause-zygote. So after that, diapause-zygote, you undergo the mitosis, right? You undergo mitosis, which is you have a cell divisions, okay? So you can produce a large number of cells. So from this ball of cell, right? From this ball of cell, you can produce a ball of cell. A ball of cell. Wait, this one, a ball of cell. Oh, sorry. A ball of cell. So this ball of cell, actually, they contain same gene. Same gene, because you also produce from that the zygote, right? The zygote, which is the diploid, the diapause-zygote. So you will just undergo mitosis, which is you make a lot of copy. You make a lot of copy, and then you become a ball of cell. So, but what at the end, right? You become the different cell. Look like different cell because you have like head parts, the eyes, the hands, the nose, right? They just look different. It's because that even though they have same genetic information, but each of the cell, right, they tend to express their genetic information in a different way. So this is, at the end, the cell will become different in their appearance, different in their functions. I'm not sure you understand what I mean. Do you understand what I mean? Or you feel very blur? Okay, so you can see all body cells in the organism, they contain same gene. So you can see same gene. Why is same gene? It's because you are from the same the zygote. Just you just undergo the cell division, right? You become a ball of cell. So just like they contain the same genetic information, but the way of their expression is different. So this is a key. I mean, this is an important point. Due to the way of expression of the genes is different. Many genes in the particular cell, right? They are not expressed because the cell only make the specific proteins in the if it needed. Okay, it means that even though they have the same genetic information inside, but not all is being expressed. They will only express if they need to make the proteins. So they only express it. And then the mitosis, okay, mitosis, right? Mitosis is just like the cell, okay? Most body cells are 2 copies of each chromosome. So just like we talk about somatic body cell, which is your skin cell, your hair cell, any kind of cell. So those are diploid because they have 2 sets, okay? They have 2 sets. So this is to ensure that when the cell split into 2, right, each new cell still have 2 copies of each chromosome, which is diploid. So which is from the diploid parent cell, they will produce the diploid daughter cell. So the purpose is they want to replace the damaged cell, yeah, for tissue growth, repair of tissue, okay, replacement of the cell, and also the essential productions like the mitosis. So that's why in the mitosis, right, you are actually produce the genetically identical daughter cell. So and also in the mitosis, you will increase genetic variations or you will reduce the genetic variation. Tell me in the mitosis.
Reduce.
Reduce, yeah, correct. It will reduce it. So that's why mitosis is not always good. This is the advantage, okay? The advantage of mitosis is because that you can produce a large number of identical cells in a very short time. So you can save a lot of resource, you can save the time. So it's very easy to be done compared to meiosis. But this advantage is you reduce the genetic variations. So it will cause what? It will cause something like your progeny, right? It will be less susceptible to the change in the environment. So everything is I already put at here. Okay, I think it's here. So this is mitosis, right? Oh, what happened? Sorry. So reduce the genetic variation. So all the progenies they produce by mitosis, right? They will develop same risk to disease, and then they can less adapt to the environmental change. Okay, mitosis. So that's all about this one. Yeah, this is showing that. Okay, another thing is, do you know why mitosis they can produce a same daughter cell with a very we can just restore their original chromosome number? Is because in the mitosis, right, you have a process known as what? DNA replications. You have DNA replication. So that's why you can produce another copy of the DNA. In the meiosis, right, you don't have this one. Okay, in the meiosis, you don't have this. In the meiosis, instead of DNA replication, right, you separate the chromosome. You separate the chromatic. So that's why from 2 become 1. Understand? Yeah, in the mitosis, right, you have this one, DNA replication. DNA replication is a process which is they can make a lot of copy of DNA. Actually, your DNA can be made a lot of copy. Okay, just like photostat machine, right? You can make a lot of photocopy of DNA in a very short time. DNA replication. So that's why you can produce a daughter cell, which is similar to your parent cell. Okay, that's all. Okay, this is just to show you that. Oh, okay, this is show you about mitosis. Okay, so you can see, this is a mitosis. Actually, we didn't learn this. Okay, we didn't learn about phase, but I just want to show you. So you can see, this is the parent cell. Okay, the parent cell. So what to do is they will do the DNA replication. So actually, the DNA replication they occur at this phase because interphase. Okay, there will be the DNA replication happen. DNA replications happen. Then they'll become what? Prophase. So you can see, they will form the. So you can see, we call as what? 2 copy of the chromosome for each. So like in this cell, right, you actually have about 2 pair of the homologous chromosome, which is this is 1 pair, right? See, this is 1 pair, and this is another pair. So it's a 2 pair, okay? 2 pair of homologous chromosome. 2 pair of homologous chromosome. So and if you align, okay, so you can see during metaphase, right, you actually align them in the center, and then in the anaphase, right, you separate them. So you can see how they separate the chromosome. There will be a spindle fibers. So at this side, right, they will require centriole. Centriole will produce spindle fiber. So what is spindle fiber? Spindle fiber is just a rope. The rope will help to separate, spill the chromosome into their sister chromatic. So that's why the rope is very important, because they need to split them. If they fail to split them, they will become non-disjunctions, which is what happened in the Down syndrome. You have one extra copy of chromosome, right? Okay, that's all. And then we talk about what? Oh, okay, the importance of the mitosis. So the importance of mitosis is, this is important, okay? Just want to produce a new cell, replace the cell. Yeah, so all of the body cell, like the skin cell, red blood cell, or even your bone cell, they will just undergo mitosis. Just want to replace the old and damaged cell. And then, okay, another one is, how about stem cell? Stem cell is found at your bone marrow. 你的骨髓, okay? There's a lot of stem cell in your bone marrow. So stem cell, what they do is, actually this stem cell, right, is an unspecialized cell, okay? So what's the function of stem cell? They just divide themselves only, that's all. Their function is they will do cell divisions. So why they need to do cell division? They do cell division is because that they need to increase the number of the cell. They just want to increase the number of cells, right? Increase number of cells. So that's why after they produce a large number of cells, so each of the cell, they will undergo specialization, or we call it differentiation. So for the stem cell, they will become what? They become red blood cell, white blood cell, bone cell, muscle cell. That's what we call it stem cell. And then meiosis, okay, they will give rise to the cell. They are genetically different to produce the gametes. So this is meiosis. Okay, so just remember that by this way, right, you are actually separate the chromosome. So that's why. So you can see, right, reduction divisions. So the chromosome number is being halved from diploid to haploid. So result in the genetically different cell. Okay, another thing is, and as well, why meiosis will produce a genetically different cell is because during meiosis, right, you're actually doing what? Okay, you actually separate the cell, okay? Sorry, not separate the cell, separate the chromosome. You separate the homologous chromosome, and then you separate the sister chromatic. So that's why the number of chromosomes is different for the parents. In parents, right, you have about 2 sets, but in the progeny, in the offspring, you only have 1 set of chromosomes. So it becomes different, okay? Genetically different cell. Yeah. So it starts with chromosome doubling themselves in the mitosis and lining up in the center of the cell. So after this happens, the cell will divide twice so that 1 copy of chromosome will pass to each of the gametes. So it becomes haploids. Okay. So another thing is, if you want to undergo meiosis, right, one type of meiosis from 1 parent cell, it will produce 4 haploid cells. Not 2, okay? And also not 2, 4. 1 parent cell becomes 4 haploid cells. 1 diploid parent cell, it will divide, okay? It will become 4 haploid cells. So actually, this is a process, okay? First, they will undergo the DNA replication also. So you can see, right, the chromosome number can be duplicated. And then they will do what? They can go to here. So they will start to separate, which is first, they will separate their homologous chromosome. So you can see, right, which is here. This is a homologous chromosome. So there will be spindle fiber at here. Okay, here is spindle fiber. Separate them. So at the end, you become like this, like this. Like this. And then you separate them again. You separate them at second stage, right? You are actually separate their sister chromatic. So here is actually you are separate their sister chromatic. You separate at here. Here. Here is a sister chromatic. I think I better write down, okay? So this division 2, right, actually is separate their sister chromatic, separate sister chromatic on the same chromosome. And the divisions 1, right, you actually separate the homologous chromosome. Separate the homologous chromosome. Okay, at that point, maybe you actually wonder why meiosis able to increase their genetic variations. Why? What factor caused them to increase the genetic variations? So it's because of this place. So you can see, right? Yeah, this place. From here. Here. You can see? Which is whenever they pair up together, right? They will be crossing over, crossing over. Which is the chromosome, right? Just imagine like this is one chromosome, this is another chromosome, they just cross over. So the section of DNA, right, they can swap between the maternal and paternal chromosomes. So they'll create a recombinant chromosome. So you can see, right, this is called recombinant chromosome because they just crossing over. So which is this chromosome, right, they will like this, okay, here and here cross, here and here cross together. So they will get different genetic combinations. So this is called different genetic combinations. So this will increase their genetic variations. Okay, so this is generally the process of the meiosis. Okay, this is just for you to have a look on it. Which is in the first phase, right? You actually separate their homologous chromosome. So in the second phase, which is in meiosis 2, you actually separate their chromatic. So from the one parent cell, you can produce 4 haploid cells. Okay, 4 haploid cells. That's all. Okay, and then yeah, so this is just to show the difference of the meiosis and the mitosis. I think we can have a look on some of the questions. Oh my god, time is almost up already. Okay, so okay, you can have a look on the question, right? Maybe I can show this one. Okay, this one. 2016, okay. The catalase is the enzyme. They can break down the hydrogen peroxide inside the cell. So the red blood cell, they will contain the catalase. So some dogs, they have an inheritance conditions in which catalase is not produced. So this condition is known as the acatalasia, okay? So it's due to the mutations in gene for the catalase. So what's gene and what is mutations? So this is gene, okay? Remember about this. This is very common to be repeat in the exam. A gene is a length of DNA. So you can see, right, length of DNA, just DNA, the short strand, the short regions, they can cause for proteins. So this is genes. Then how about mutations? Okay, mutation is due to what? Actually, there's 2 types of mutation, okay? We call it chromosomal mutation or gene mutation. So the gene mutation, right, is referred to any change in the sequence of DNA. Because if you cause any change in sequence of DNA, even though it's just one base, the change in one base, right, you will still induce change in the amino acid sequence later. So if you change the amino acid sequence later, right, you can change what? You can change the protein structures, and then it will affect the functions. So here's the answer. And then for this one, right, okay, this one, okay, I think we can do more in next class, right? So I just briefly tell you how to do it. Okay, first, you need to identify which is the big capital, which one is small capital, okay? Yeah, they tell you that the large capital B, right, is normal. So you can write like this, right? Let B, okay, become normal alleles, which is in the normal dots, okay, in the normal dots. Then how about small b? Okay, let small b become what? Become the mutants allele. So and then now it's time for you to write down the genotype. Okay, you should write the genotype. So you have what? You have the male and female, right? So the shaded part would be the disease, okay, the acatalasia. So the male is right. How you write the male? Okay, the male. And then one thing is, I forget to tell you, okay, you should be able to identify whether it's related to sex-linked disease or it's not sex-linked disease. So like in this case, it is definitely not sex-linked disease. Okay, what I mean by sex-linked or not sex-linked disease is like sex-linked disease. It refers to this disease, right? They carry the defects on the sex-linked gene, which is the X or Y. Okay, but in this will be the autosomal disorder. Okay, so number 2, right? Since you have the acatalasia, so it's like a small b. Okay, this one is small b. Then how about this 5? The 3 is the bb. Then number 1 is about, maybe you can write, is about bb also. So how you can make a conclusion is because like this. Okay, when you make this 2 with 3, right? So you can do by yourself. So okay, bb cross the bb, so you can produce the offspring. So it will be b, right? bb, and then b, bb, and bb. So it's about half of them will be the normals, but it's carrier, because it's large B and one small b, right? So it's actually it's carrier, even though it's normal. And here will be affected. So that's why 5, right? 5 showing that it is small b. And then here showing what? 1b, 1 small b, 1 bb. Same to here also. So what happens if number 1 is carrier? So if number 1 is carrier, right? Then here also carrier. What will happen is bb become bb. Okay, and then bb and bb. So you will have one of the progeny is defects. Okay, and then other one will be normal, but there's 2 of them will be carrier. So this is how you're able to guess it. Okay, just to guess it. Okay, another one is, what's the time now? Okay, 24. It's okay. I think I will discuss more in next class. So just want to highlight to you some of the important point for this chapter, right? Okay, this chapter, I think I start to talk about here. This one. This one, okay? Here. So we can see, right, the DNA-based sequence, how they can determine the amino acid sequence, the structure of proteins, type of protein, and then protein functions. And then you have what? Okay, you have the homozygous. What do you mean by homozygous? Homozygous is they refer to same. Homo means same. Hetero means difference. Difference in their genotype. So if homo, you have homozygous dominant or homozygous recessive. So homozygous dominant means it's both genotype, right? Because one gene, at least they carry 2 alleles. One gene, they must have 2 alleles. So that's why both of the alleles must be dominant, which is big B. Okay, if recessive, right, both of the alleles must be small b. Okay, small b. Heterozygous refer to one big B, one small b. And then, yeah, one genotype, right, they must have 2 alleles. 2 alleles. So what's my alleles? Alleles is referred to the alternative version of the genes. So it means what? It means that alleles, alternative version of genes means that they carry same gene functions, but they express genes in different physical features. For example, like one hair color gene, right? You can have different color, okay? You can have yellow color, brown color, black color, but they are all belong to same gene, which is they're just having the hair color gene, which is they carry the same functions. Yeah. And then if you want to refer to the nuclear base sequence, right? your sequence is always very similar to your parents, okay? Or your but it's a bit different compared to your friends or your relative. So this is called sequence. And then one thing is, what's about test cross? Okay, test cross, right, is a kind of experiment cross of the individual organisms of dominant phenotype with unknown genotype. So you can see, right, one dominance with the unknown genotype, because they want to test that whether this phenotype, right, is due to the homozygous dominant or heterozygous dominance. This is their purpose. They want to find out which one is heterozygous, which one is which one is homozygous. So that's why they do this test cross. Test cross must be crossed with the homozygous recessive. So you can see, right, this is test cross. So in the first test cross will be the purple flower with the white flower. Second will also be the purple flower and white flower, but you are not sure. You are not sure this purple flower, right, is homozygous or is heterozygous. So that's why it must be crossed with the white flower first, which is both of them are small b. So you have the heterozygous, right? One big B, one small b, and two big B. So what happened is after you cross, and then you produce the gametes and the offspring. So you have what? You have the purple flower and white flower. Means that you will have about 50% is belong to purple flower, 50% is belong to white flower. So from this method, right, you can identify that the parents is belong to heterozygous dominance, but not homozygous dominant. It's because that if you cross this homozygous recessive genotype with the homozygous dominance, right, you will get all is belong to purple flower. None of them is white flower. So by this, right, you can decide that, oh, this is homozygous parents. Okay, so this will mean that test cross. Just want to decide whether it's heterozygous or homozygous dominant. So that they need to cross with the homozygous recessive genotype. Okay, and then okay, sickle cell anemia, I think we can discuss in next class. Okay. I just briefly tell you about how to do the genetic question. I think you can go back to try some of the question. Or if you don't want to try, it's okay, because we will do together in next class, okay? Because some of the question is quite difficult for you, okay? And then I just want to let you know is, if you want to do this kind of genetic question, right, there's some step, you know, the procedure. So I have write down the procedure at this document. First, you need to identify whether this question, right, is sex-linked or autosomal disease. Sex-linked means what? Means that there's a kind of disease. The disease is located on the sex chromosome, which is usually is located on the X chromosome. So you can see, right, X chromosome is because the X chromosome is always larger in size. So we call it sex-linked disease. Autosomal disease refer to this disease, right? It's not located on the sex chromosome, which is X or Y. No, they will just only located on either from the chromosome number 1 to number 22 pairs, either one of chromosome. So this is called autosomal disease. So before you do the genetic question, right, you need to identify whether it's autosomal disease or it is a sex-linked disease. Okay, so how you can able to identify is you can read on the question. Okay, if you see that the question they're talking about, like the male, usually is all male being affected, and then female is a carrier. So that's why the question might be talking about sex-linked disease. Because if you talk about autosomal disease, right, no matter it's male or female, all of them are being equally being affected. Okay, all of them will be equally being affected by the genetic disease. Because in the sex-linked, right, usually only male is being affected. Female is a carrier. It's because that male, right, male, they have X and Y. XY, okay? Yeah, chromosome is XY. So it's because that they only have one X. So if the X chromosome, they contain the defect genes, they will just show the, they'll show the symptom. In the female, right, it's always carrier. It's always a carrier. It's because female, unless both of the sex chromosome, which is the X chromosomes, carry the mutant alleles. they will only show the, they will only show the sign. But if the female, right, they have only one X chromosome, carry the mutant alleles, but other X chromosome is normal. They will not show the sign, but they will pass on these mutant alleles to the next generations. So usually the female is a carrier. Carrier means what? Carrier means that they carry the defective genes, but they will not show the sign. They will not show any symptom, but they will transfer these defects to the next generations. And then usually male will be the affected one. Okay, so that's all for the class today. Yeah, we will continue in next class. So I hope you understand more about the inheritance. Okay, it's very interesting topic. Okay, see you. Bye.